MS Ramaiah University of Applied Sciences.
Determination of blood group. Aim of the experiment: to determine the blood group of the blood sample. Introduction. Blood transfusion is a life-saving procedure in all cases of severe loss of blood and in life-threatening anemias. However, blood can only be given after blood grouping, which is an essential requirement before blood is given to any individual. Blood grouping is also done to settle paternity disputes and other medico-legal purposes. Experimental setup: compound microscope, clean and dry microscopic slides, blood lancet, sterile cotton swab, 70% alcohol, toothpicks, glass dropper, saline and antisera. Experimental procedure: using a glass marking pencil, mark 3 slides as A, B and D respectively. Divide the slides into 2 halves. The left half serves as a control side and the right half serves as test side. Preparation of red cell suspension: place 8 to 10 drops of saline on another slide named S. Get a sterile finger prick, add a drop of blood to the saline. Using a toothpick, mix well to get a red cell suspension. Place normal saline on left half of the slides A, B and D, which serves as control. Place antiserum on the right half of the slides, antisera A to slide A, antisera B to slide B and antisera D to slide D. Add a drop of red cell suspension to all the slides. Use separate toothpicks and mix it. Wait for 8 to 10 minutes, then inspect the mixtures first by direct vision and then through the low magnification microscope. Express your results according to presence or absence of agglutination. Analysis: surface of red cell membrane contains a variety of genetically determined antigens called isoantigens or agglutinogens, while the plasma contains antibodies called agglutinins. To determine the blood group of a person, the red cells are made to react with commercially available antisera containing agglutinins. The slide is then examined under microscope to detect the presence or absence of clumping and hemolysis of red cells, which occurs as a result of antigen antibody reaction. Precautions: the slides should be dry, clean and grease free. Double check every step of the procedure. The droppers supplied with antisera bottles should not be interchanged. Examine the slide after 8 to 10 minutes, but before the antisera blood mixtures dry up. Do not add the undiluted blood from the finger prick directly to the antisera to avoid false positive reaction.
Hi students, today we are going to study determination of blood group. The aim of this experiment is to determine the blood group of given blood sample. Blood transfusion is an essential life-saving process in all severe cases of traumas, injuries or even in case of severe anemias. Once there is more than 35% blood loss from the body, it will lead to severe physiological changes which may eventually lead to the death of the person. Hence, the blood transfusion plays a very essential role in retaining the normal physiology as well as in the saving of the life of a person. However, the blood can be only given after performing the blood grouping experiment, which is an essential requirement to avoid mismatching before the blood group is given to any individual. Blood grouping is also done to settle paternity disputes and other medico-legal purposes. Now, let us study the different requirements used in this experiment. This experiment requires Lancet device. Normal saline. Normal saline is used to dilute the blood. Dry cotton swab to remove the excess of alcohol. Cotton swab with 70% alcohol. Antisera A, Antisera B, Antisera D. Antisera A, which contains monoclonal anti-A antibodies. Antisera B, which contains monoclonal anti-B antibodies. Antisera D, which contains monoclonal anti-RH antibodies. The experiment is based on the principle of reaction between antigen and these monoclonal antibodies, and glass slides with the following marking: A, B, D and S, and Pasteur pipette. Now let us start the experiment. First, sterilize the finger bulb with 70% alcohol containing cotton swab thoroughly on both the sides and allow the alcohol to evaporate. Then, using the lancet device, take a bold prick and discard or remove the first drop of blood using the dry cotton swab, as it may contain tissue fluids. Then add 2 drops of the blood on the slide named with S. After adding 2 drops of blood, add 7 to 8 drops of normal saline. And using one more glass slide, mix the blood with the saline. This diluted blood will be utilized for further experiments. Then the diluted blood with the saline is added to the A slide, B slide and the D slide. Then the antisera A is added to the slide named as A. Antisera B is added to the slide B, and antisera D is added to the antisera to the slide named D. Then, using one more glass slide, all the 3 different slides containing diluted blood and the respective antiseras are mixed using the different corners of the glass slide. And after thoroughly mixing, the slides are observed for antigen antibody reaction, which will lead to agglutination. If there is agglutination in slide A, then the blood group is A. If there is agglutination in slide B, then the blood group is B. If there is agglutination in both slide A and B, then the blood group is AB. And if there is no agglutination in slide A and B, then the blood type is O. If there is agglutination in slide D, then the blood is said to have Rh positive factor, and if there is no agglutination in the slide D, then the blood is said to contain Rh negative factor. In this case, there is no agglutination or clumping in case of slide A and in case of slide B. Hence, the blood type is O, and there is agglutination or clumping of the blood in case of slide D. Hence, it can be concluded that the given blood sample was O positive. This completes the determination of blood group experiment.
At MSR UAS, we spark your imagination unlike any other.